Imagine the human brain as like an ultra-exclusive VIP club.
Right.
It has the absolute strictest bouncer in the physiological world standing right at the door, the blood-brain barrier.
Oh, definitely. The ultimate velvet rope.
Exactly. And for decades, the fundamental consensus in biology was just that no unauthorized guests could ever get past that bouncer. I mean, no microbes, no intruders, just a completely pristine, sterile environment of neural real estate.
Right. That was textbook neuro-oncology for, well, forever.
But a mind-bending November 2025 paper from Nature Cancer just completely shattered that illusion. It turns out there is a secret party happening right inside brain tumors, and bacteria are not only on the guest list, they might actually be running the show.
Yeah, it really forces a total rewrite of what we know. I mean, we are looking at thriving, highly complex microbial ecosystems operating inside a place we historically treated as an impenetrable fortress.
Okay, let's unpack this. Because today we are taking a deep dive into the microscopic trenches of this discovery, we're going to figure out how these bacteria bypass the bouncer, where they set up shop inside your brain, and most importantly, how they're actively manipulating cancer treatments from the inside out.
It's a wild ride.
It really is. Have you ever considered that a tumor in your brain might have its own thriving bacterial ecosystem? But before we get into the how, we have to address the elephant in the room. If I'm a skeptical scientist reading this, my immediate pushback is, well, operating rooms are not vacuum sealed, right? How do we know a researcher didn't just, breathe on a petri dish?
Right, contamination. It is the ghost that haunts literally all low biomass microbiome research.
Oh, I bet.
And the researchers knew they would face that exact skepticism. So they didn't just rely on one test, they built this multi-layered 6-method detective protocol to prove these bacteria are native physical residents of the tumor. First, they had to actually visualize them in the tissue using a combination of immunofluorescence and something called RNA F/H.
Wait, okay, let's pause and explain those mechanisms for a second, because we can't just throw acronyms around. If I want to see a microscopic bacterium hiding inside a human brain, how does RNA F/H actually light it up?
So think of RNA F/H like sending millions of microscopic, glowing, homing beacons into the tissue.
Okay.
A FISH stands for fluorescence in situ hybridization. These probes are engineered to bind exclusively to universal 16S ribosomal RNA, which is basically a genetic signature that is completely unique to bacteria.
Got it. So human cells don't have it.
Exactly. When they find that bacterial RNA, they latch on and they literally glow under a microscope. And they pair this with antibodies targeting EF2.
EF2, what's that?
It's a highly conserved protein. Think of it like a universal uniform that almost all bacteria wear.
Okay, so you have these glowing beacons lighting up the bacterial uniform. Where exactly in the brain tissue did you see them? Are they just like floating around in the extracellular fluid between the cells?
That is where the imaging gets genuinely stunning. They are not floating in the void. They are entirely intracellular.
Wait, inside the human cells?
Yes. The researchers found them physically hiding inside the human cells. They tracked them inside cytokeratin and GFAP-positive cells, which, proves they are inside the actual tumor cells, not just the surrounding tissue.
Wow.
And even crazier, they found them inside CD68-positive macrophages.
Hold on, macrophages are immune cells? I mean, their entire job in the body is to eat and destroy bacteria?
Right. Yes, and yet here the bacteria are, surviving inside the very cells meant to kill them. It's a literal Trojan horse scenario. Visually, these bacteria were nestled right up against the nuclear membrane of the host cells.
That is a terrifying level of intimacy. I mean, they are practically hugging the human DNA. But a glowing dot on a screen is one thing. How do you prove it's a physical, structured bacterium and not just some stray clump of proteins that happen to bind to the probe?
For definitive structural proof, they brought in transmission electron microscopy, or TEM, and they combined that with anti-LPS gold labeling.
Okay, break that down for us.
So LPS, or lipopolysaccharide, is a major structural component of the bacterial outer membrane. The researchers attached these tiny, heavy gold particles to antibodies that specifically hunt for LPS.
Oh, I see. It's like dusting a crime scene for microscopic fingerprints, but using gold dust that only sticks if a bacterium physically touched that exact area.
That's a perfect analogy.
Yeah.
And the electron microscope captures the exact physical architecture of those gold-dusted bacteria embedded deep inside freshly resected tumor tissue.
Incredible.
But, structural proof still isn't enough. You could have a dead, fossilized bacterium sitting in a tumor. The researchers needed proof of life. They needed to show these microbes were actively metabolizing.
Right, which brings us to culturomics. I know they took fresh glioblastoma tissue and literally tried to grow the bacteria in the lab across, 32 different types of agar?
Yeah, 32 types, both with and without oxygen.
Hmm.
But the fifth method, the dealanine test, is the one that feels like pure biological engineering.
Yes. Walk us through how they used an amino acid to prove the bacteria we're actually eating.
So this relies on a really beautiful quirk of evolution. Mammalian cells, like our human cells, exclusively use the L structural version of amino acids to build proteins. They completely ignore the mirror image D versions.
Okay, so they're like left-handed and right-handed molecules.
Exactly. But bacteria rely heavily on dealanine to build their cell walls, specifically this layer called the peptidoglycan layer. So the researchers introduced fluorescently labeled dealanine to fresh tumor slices.
Oh, wow. Because the human cancer cells physically cannot process dealanine, the only way that tissue is going to glow is if something else inside is hungry for it.
And the tumors lit up brilliantly. The live, metabolically active bacteria were actively consuming the dealanine to rebuild their cell walls right there in real time.
That is so smart.
And to verify this, they pre-treated some tumor slices with levofloxacin, which is a broad-spectrum antibiotic. The fluorescence completely dropped off, confirming the signal was driven by living bacteria that the antibiotic had just killed.
Makes total sense.
Finally, they used 5R16S rDNA sequencing to map the exact genetic identities of these survivors.
Okay, but we still haven't fully addressed the operating room error. Even if they are alive and eating, maybe they just fell into the brain while the patient's skull was open.
The researchers anticipated that too.
Yeah.
They ran 468 negative controls.
468.
Yep. They literally opened blank tubes to the air in the operating room during the neurosurgeries and processed them identically alongside the tumor samples.
And what did they find?
Out of all those controls, they only grew 37 colonies. 4 were typical environmental contaminants, like Staphylococcus epidermidis. But the remaining 33 colonies, including species like Acinetobacter baumannii, were uniquely native to the tumors. The mathematical and biological controls definitively ruled out surgical contamination.
Okay, let's pause and summarize what we know so far for you, the listener, because that was a lot of dense science.
Exactly.
Bullet 1. The brain is not sterile. We've fundamentally broken that myth. Tumors host intracellular bacteria. Bullet 2. We know they are alive because they eat bacterial-specific food, like dealanine, and can be cultured in a lab. And bullet 3. Strict exhaustive controls prove this isn't just lab or surgical contamination.
Exactly.
So imagine tiny bacteria living right next to the nucleus of your cells. What do you think they are whispering to the cell's DNA?
That intimate proximity changes everything. Because once you accept they are setting up shop in the brain, the next logical question the researchers asked was about real estate.
Right. Does a tumor that originates in the brain attract the same bacterial guests as a tumor that invades from somewhere else?
Exactly.
This is where we get into a tale of 2 distinct microbiomes. The paper compares glioblastoma, which is a primary tumor born and raised in the brain, against brain metastases, which are secondary tumors. You know, cancers that started in the lungs or the breast, mutated and broke into the brain.
And the data reveals a stark, almost algorithmic contrast in biodiversity. Brain metastases are significantly more microbially rich and diverse than primary glioblastomas.
How much more diverse are we talking?
To give you the specific clinical breakdown, a standard glioblastoma sample averages about 7.8 different bacterial species. But a brain metastasis originating from breast cancer averages 12.5 species, and a lung cancer metastasis averages 13.5.
Okay, I have a mnemonic device for the listener to remember this. Think of glioblastoma, GBM, as a grounded, basic municipality. It has fewer species, it's a bit more isolated, while metastases are massive, bustling metropolises, highly diverse, lots of different inhabitants.
I love that. The ecological analogy holds up perfectly when you look at the actual sequencing. They don't just have more bacteria, they have entirely different families.
Give us some examples.
Sure. So GBM is dominated by the Rhizobiales order, like Bartonella jaculi. But the metastatic tumors are enriched with entirely different populations. You see lactobacillus, such as Carnobacterium viridans, and Burkholderioles, like Rubervivax.
Wow, okay. But this introduces a massive mechanical puzzle. If I have a tumor in my lung, and months later a secondary tumor appears inside the blood-brain barrier, how did lung-specific bacteria get past the bouncer? Did they just happen to wander into the brain independently?
What's fascinating here is The sequencing data strongly suggests they did not wander independently. The researchers compared the microbiome of these brain metastases directly to the microbiomes of independent primary breast and lung tumors.
And
They found that 36% of the bacterial genera in the breast metastases and an astonishing 48% in the lung metastases were perfect matches to the primary tumor sites.
wait, almost half. So they packed their bags and traveled together. We are looking at a literal hitchhiker mechanism.
It appears so. The bacteria are physically traveling with the circulating tumor cells right through the bloodstream.
But the bloodstream is an incredibly hostile environment. You have intense fluid shear forces that want to rip cells apart, and a continuous patrol of white blood cells hunting for foreign invaders. How does a cancer cell, let alone a bacterium, survive that transit?
That is the crucial insight. We've actually seen this dynamic in colorectal cancer, where bacteria like Fusobacterium nucleatum travel to the liver. This paper extends that paradigm to the brain.
So they're helping each other.
Exactly. The bacteria might not just be passive passengers, they could be acting as armored escorts. By binding to the cancer cells, the bacteria might alter the physical stress tolerance of the tumor cell, or secrete proteins that blind the immune system, actively helping the cancer survive the journey and breach the blood-brain barrier.
Time for another quick checkpoint to consolidate this layer of the puzzle. Bullet 1. Brain metastases are much more microbially diverse than primary glioblastomas. Bullet 2. Different tumors attract entirely different bacterial families. And bullet 3. Metastatic bacteria likely hitchhike from the original lung or breast tumor into the brain.
Spot on.
So if bacteria are hitching a ride with cancer cells and acting as bodyguards, could we one day use targeted antibiotics to block cancer from spreading? It's a huge question to think about.
It's a multi-million-dollar clinical question, truly. But the researchers didn't stop at tracking where the bacteria came from. They wanted to know where they ultimately settled. They mapped the bacteria by physical coordinates inside the brain itself.
Here's where it gets really interesting. We've covered the who and the how, but the where is completely fascinating. They mapped the brain's bacterial geography and found a strict division based on the anterior-posterior axis.
Right, the front of the brain versus the back of the brain.
Exactly.
The data showed that bacterial richness significantly rises as you move toward the back of the skull. Tumors located in posterior regions like the cerebellum and the occipital lobe have far greater bacterial diversity than tumors located in the anterior regions like the frontal lobe.
But why would a bacterium care if it's sitting behind your forehead or near the base of your skull? I mean, it's all brain tissue, isn't it?
It is, but the microenvironments are distinct. And this specific bacteria setting up shop in the back of the brain taxa, like Salmonella, Bacillus, and Enterococcus, they are not just randomly accumulating. The researchers ran the genomic data through a computational tool called PCE-C2 pathway analysis.
Okay, let's demystify that algorithm. What is PCE-RIS2? How does a computer program tell us what bacteria are doing inside the cerebellum?
Think of it like reading a blueprint. PCE-RIS2 takes the genetic sequencing of the bacteria we found and references a massive database of known biological functions. It essentially predicts what metabolic factories those bacteria are operating based on their DNA.
Oh, that's clever.
And the algorithm revealed something amazing. The bacteria in the posterior brain are heavily enriched with genetic pathways to synthesize vitamin B1, which is thiamine, and vitamin B7, biotin.
Wait, really? They are actively manufacturing highly biologically active vitamins inside the tumor. Both B1 and B7 are critical for cellular metabolism and energy production. The cancer cells must be absolutely thriving on that extra energy supply. It's like finding out the back rooms of a house have a completely different microclimate and garden than the front yard. But why? Why are these specific vitamin factories only being built in the back of the brain?
The mechanism almost certainly comes down to the brain's anatomical plumbing. The front and the back of the brain are fed by entirely separate arterial highways.
Ah, the blood supply.
Exactly. The anterior brain is primarily supplied by the internal carotid arteries, while the posterior brain gets its blood from the vertebral arteries.
So different blood vessels might literally be sweeping different bacterial cargo from the rest of the body into different neighborhoods of the brain.
Yes, or the blood from the vertebral arteries provides a slightly different mixture of oxygen and nutrients that allows the Bacillus and Enterococcus to thrive there specifically. It forces us to view the brain not as a single uniform organ, but as a diverse topographical map with distinct ecological niches.
Let's consolidate this map for you. Bullet 1. The back of the brain hosts a richer, more complex bacterial community than the front. Bullet 2. These posterior bacteria are actively producing vitamins like B1 and B7 and bullet 3. This geographical divide might be due to the brain's distinct arterial highways.
Exactly right.
Think about the anatomy of your own brain right now. Isn't it wild that different bacterial communities prefer different neighborhoods up there?
It is biologically awe-inspiring. But, as this is nature cancer, we have to bridge this incredible biology to the actual patient lying in the hospital bed.
Yeah.
Because ultimately, we need to know if all this bacterial mapping actually impacts human survival.
Right. All these maps and mechanisms are fascinating, but what are the clinical stakes?
Yeah.
And the data they abstracted is incredibly sobering.
It is. When they correlated the microbiome data with clinical outcomes for patients with brain metastases, a harsh algorithmic threshold emerged. High bacterial richness directly equal lower overall survival.
Let's make sure that is perfectly clear. The more diverse the bacterial ecosystem inside your brain metastasis, the shorter your life expectancy.
Specifically, they found that patients whose overall survival was less than 1 year had significantly higher bacterial richness. And they saw a very specific enrichment of a bacterial genus called Paracoccus when compared to patients who survived more than 2 years.
And did they see that in glioblastoma too?
Interestingly, no. They did not find this same harsh survival correlation in GBM, which once again highlights how mechanistically different primary and secondary tumors are.
But anytime we look at survival data, I have to play devil's advocate. Correlation does not equal causation. I mean, if a patient is closer to death, their immune system is likely failing, right? Which might just allow more bacteria to grow. How do we know the bacteria are actively making the cancer deadlier rather than just taking advantage of a dying patient?
That is the exact right question to ask. To answer that, they had to isolate the variables. They moved from clinical observation back into the laboratory for an in vitro case study.
Okay, what did they do?
They took human glioma cancer cell lines, specifically the T98G and U251 lines, and grew them in controlled culture dishes. Then, they exposed these cancer cells to a common chemotherapy drug called temozolomide, or TMZ.
TMZ is a heavy-hitting frontline chemo drug. Its entire mechanism of action is to induce catastrophic DNA damage and force the brain cancer cells to self-destruct.
And under standard conditions, the TMZ does exactly that. But the researchers introduced a twist. They added bacterial preconditioned media, or PCM, to the cancer cells before giving them the chemo.
For those trying to visualize PCM, think of it as the bathwater that the bacteria had been living and growing in. It doesn't necessarily contain the physical bacteria themselves, but it is super saturated with all the metabolites, proteins, and chemical signals those bacteria secrete.
Exactly, the bathwater. They specifically used the secretory bathwater from the Bacillus genus, which they knew was highly prevalent in these tumors.
And what happened?
When the cancer cells were bathed in these bacterial secretions, they suddenly became highly resistant to the chemotherapy. The TMZ could no longer efficiently kill them.
Wait, so it's not the physical bulk of the bacteria blocking the drug, it's the invisible chemical soup they leave behind. So what does this all mean? How does that chemical shield actually work?
If we connect this to the bigger picture, the bacteria aren't just innocent bystanders. They are actively changing the tumor's microenvironment.
Are there secretions acting like a sponge to absorb the drug?
It could be that the bacterial metabolites physically bind to and degrade the chemo agent before it reaches the nucleus. Or they might trigger signaling pathways within the human cancer cell that upregulate its own DNA repair mechanisms, effectively neutralizing the damage the TMZ is trying to cause. Either way, the bacteria are actively manufacturing a biochemical shield, potentially shielding the cancer from our best drugs.
We have gone on an intense paradigm-shifting journey today. Thank you for sticking with us through the dense molecular biology. We started by definitively breaking a decades-old myth, proving with exhaustive, multi-layered detective work that metabolically active bacteria do, in fact, live inside brain tumor cells.
Yes, we did.
We tracked their hitchhiking journeys from the lungs and breast, surviving the bloodstream to bypass the blood-brain barrier. We mapped out their preferred neighborhoods in the posterior brain, where they act as vitamin factories. And finally, we saw the terrifying clinical reality of how their chemical secretions can physically intercept and block chemotherapy from saving a patient's life.
The implications for the future of oncology are monumental. It demands a complete recalibration of how we treat the tumor microenvironment.
Which leaves me with a final lingering question for you to mull over. We've talked extensively about how these bacteria build chemical shields, and earlier we discussed how they rely on highly specific nutrients like D-alanine to survive and build their cell walls. If cancer treatments are failing because bacteria are protecting the tumors as bodyguards, are we fighting the wrong enemy? I mean, could the future of brain cancer treatment lie in manipulating the gut microbiome or utilizing highly targeted dietary fasting to literally starve the brain tumor's bacterial bodyguards of their specific amino acids before we ever administer the chemotherapy? In the future, before we give a patient chemo, will we have to give them a highly specific antibiotic first?
If we can collapse the bacterial supply chain through diet or gut manipulation, the tumor shield falls. It is a brilliant avenue for the next wave of research.
Thank you for joining us on this deep dive. Keep questioning the established science, stay curious, and remember, the VIP club in your head might just have a few unauthorized guests pulling the strings. Catch you next time.